All right, so here we are going to talk about genes, chromosomes, and cells. And so the first I can statement is I can define a gene. A gene is simply a segment of a chromosome. So if this is we'll make him kind of chunky so you can see a little bit better. Here's our chromosome. There's our centromere in the middle. Somewhere on this chromosome, there is a section of DNA. A sequence of letters. It can be anywhere from a couple hundred base pairs to a couple thousand. It just depends. That codes for a specific protein. And so that's all a gene is. It is a segment or section of a chromosome that has a start and stop codon and built into it so that the cell knows when to start and stop transcribing it, and it will code for one protein or one mRNA. Some of those mRNAs become proteins and others don't. So a gene is just a section of chromosome that codes for an mRNA. The second is I can explain what it means that a gene is found on a specific locus on a chromosome. Okay, let's say this is chromosome number 1. Okay, you have 23 pairs of chromosomes, so 46 total. Chromosome number 1 would be that biggest pair. They're always arranged from largest to smallest when you do a karyotype, so chromosome number 1 is just the biggest one. If this particular gene is found on this arm, which maybe is a little bit longer, I can't tell. But let's say this is the long arm. Most chromosomes have a long arm and a short arm. So if this is found on the distal end of the long arm of chromosome number 1, then it's going to be found on the distal end of the long arm of chromosome number 1 in every human on Earth, unless there's been some kind of translocation or other mutation. And so that's what it means. It's found in a specific location or geography on that chromosome, and it's the same for every human. Other species would have different genes in different loci, but every gene has its own specific location within that species where it's found. I can define homologous chromosomes. All right, so let's say this is the chromosome you got from your dad. So you got 2 chromosome number 1s, okay? One from mom, one from dad. And the chances of me drawing them the same are slim, but I will try. Okay, so there's your chromosome number 1. It does look a little different, but pretend it looks the same for mom. Homologous chromosomes are chromosomes that have all the same genes in all the same places. So if this is the gene, let's say this is the gene that allows you to roll your tongue or not, you're going to have a gene from your mom in the same location on her chromosome for the same thing. So they are just homologous chromosomes. Homo means the same. And so they are just chromosomes that have all the same genes in all the same places. And so each pair of your chromosomes are going to be homologous. So chromosome set number 1 are homologous. Chromosome number 2 is homologous with the other. So you'll have a dad chromosome number 2 and a mom chromosome number 2, and they'll have all the same genes in all the same places. So you have 22 sets of homologous chromosomes, and if you're female, you have 23 sets of homologous chromosomes because your X chromosomes are homologous as well. If you are a male, you have an X and a Y, they're not homologous because the X is much larger than the Y and it has different genes than the Y. So if you're a male, you have 22 pairs of homologous chromosomes and then your X and Y chromosome. Let's see, I can compare autosomes and sex chromosomes. Well, both of them are chromosomes. Both of them come in pairs. So you get one from mom, one from dad. So you have 2, one from mom, one from dad. Autosomes are, well, let's start with this. Sex chromosomes are going to contain the genes that determine your gender, your biological gender. That determine biogender. Okay, so they're going to have the genes that code for the development of either ovaries or testes. Autosomes are going to have genes not related to gender. Now, I say that there are some genes on the X chromosome that aren't related to gender. There are some, there's one that has to do with the rods and cones in your eyes, and that's why there is an X-linked color blindness. There's one that has to do with muscle development. That's why there's an X-linked muscular dystrophy. There is one would do with blood clotting, so there is an X-linked type of hemophilia. So there are other genes on the X chromosome, but what determines a sex chromosome, which would be the X or the Y, is that it does contain the genes that determine gender, whereas the autosomes have all the other genes. All right, so you have 22 pairs of autosomes and 1 pair of sex chromosomes. All right, so that's comparing and contrasting those. I can compare and contrast haploid and diploid cells, okay? Well, they are both cells. They both have DNA, genetic material, whatever you want to call it. You could go into a lot of detail here. They both have nucleus. They both have mitochondria, Golgi, rough and smooth ER, lysosome. I mean, you could list all kinds of things that they both have. But what we're really focused on here is the genetics. So a haploid is going to have one copy of each chromosome. We call that our N number. So if you ever see N, that's just talking about haploid. And for humans, haploid means you have 23 chromosomes. This is what you're going to see at the end of meiosis. And you're going to see this with your gametes, which is just a fancy word, whoops, for sex cells, sperm and egg. Okay. Diploid is going to have two copies of each chromosome. Okay, so you'll see that abbreviated as 2N. For humans, that is, of course, 46. You are going to see diploid at the beginning of both mitosis and meiosis. You're going to see diploid at the end of mitosis. And you're going to see this in your somatic cells. You also would only see haploid in sexual reproduction, where you would see diploid in both sexual and asexual. So those are just some of the ways that they're alike and the ways that they're different. So that's comparing and contrasting. And the last is I can explain how karyotypes are used to analyze chromosomes. And the short answer to that is a karyotype is simply a snapshot of the chromosomes in mitosis, because that's when they're condensed and they're easy to see. Generally, they take a picture of them in late prophase or in metaphase, because they're generally kind of near the center of the cell then, so they're easy to get pictures of. They're not all scattered all over the place. Oh, sorry. Let me cover that light up. And a karyotype is going to show you the chromosomes, and it's going to allow you to count them, so you can make sure there are indeed 46. There are no extras or none missing. It also will allow you to compare them, so you can compare, like the dad, you don't know which one's dad and mom, but you can compare the two homologous chromosomes for each pair and notice any size differences. If there's any that are broken or missing pieces, you can do that. So it's a really good tool to use to diagnose chromosomal abnormalities, large chromosomal abnormalities that are visible when you look at the chromosomes. And so that's what they're used for. And that's it for these I can statements. If you've got any questions, let me know.